Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, patient advocate Donna Cryer, liver wellness advocate Louise Campbell, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on the Surfing the NASH Tsunami podcast.
For everyone with an interest in NASH or, more broadly, fatty liver disease, Surf's Up, episode 43 of Surfing the NASH Tsunami starts now. This is Roger Green, executive producer and host of Surfing the NASH Tsunami, and I want to welcome you to our 2 year-end episodes, the first of which airs today. We invited 5 of our favorite guest surfers, Vlad Ratziu, Bjorn Schottenberg, Mazen Noureddin, Ian Rowe, and Wayne Eskridge, to sit for a 20 to 25-minute interview focusing on what each considered an important trend or issue in fatty liver disease in 2020. Louise, Stephen, and I each contributed our own segments. Louise and I participated in all 5 interviews with the guests. Stephen joined as his schedule allowed. This is hardly the complete list of people we would have liked to include. Donna Cryer gave us her time for the last 2 episodes of the year, and as a result had none left before her calendar closed up for vacation. Others originally thought they could attend, but had to cancel at the last minute for a variety of reasons. But what this group lacks in gender diversity, it compensates in diversity of issues, as you will hear. We gave no guidance, but each participant came up with a unique issue to address. Half of these could loosely fall under the heading Drugs and Diagnostics, while the other half could loosely fall under the heading Patient Treatment and Perspectives. This week, we present the Drugs and Diagnostics interviews. Next week, episode 44, we'll present the Patient Treatment and Perspectives interviews. I will provide a brief introduction to each interview, and then we will dive right in. No icebreakers, no chit-chat, minimal to no preliminary conversation. We have a lot to cover, not a ton of time. Before we start, a point about the COVID-19 pandemic. Everything this year and much of what we discuss today and next week occurs against that backdrop. So first, I want to salute all the healthcare professionals who've not only done their jobs, but have also gone to the front lines and helped colleague health professionals in the fight against this pandemic. And then second, I hope everybody is bearing up okay under whatever constraints COVID is placing on your holiday this year. We have vaccines starting to roll out. If we can get through the next few months, things should get better. So let's all hang in there, do what we got to do, and get to the other side of this quickly. With that said, on to the interviews. Our first interview is with Vlad Ratziu, who joined us for EASL and AASLD wrap-ups and is a good friend of the podcast. He will present a dazzlingly fast and thorough summary of major moments in diagnostics and drug development in the year 2020. This conversation is with Vlad Ratziu, who's joined us for our EASL and AASLD coverage and once or twice more throughout the year. And Vlad, welcome. And which event of 2020 would you like to focus on today?
So thanks for the invitation, Roger. It's always a pleasure to be here and discuss with you all. So obviously 2020 was a very special year with this pandemic that hit all of us and had a profound impact on both our professional activities and on the way we interacted with each other. And there's a lot to say about this, but obviously what we need to focus is on, in terms of NASH landscape, what happened during this year. So despite the bad things that happened, the limitations in terms of conducting clinical trials, this was a very good year actually for NASH clinical trials. I think that one of the major things that 2020 brought us is some significant advances in terms of therapeutics for NASH. We had the results of several trials this year. We had the unfolding story of other molecules that are in development for NASH. And all this makes for a very, very vibrant environment in terms of therapeutic research in NASH. So I'm very pleased about that. We had both results from phase 3 trials and results from early phase, smaller proof of principle trials with innovative molecules, both reported in the same year. So the field is clearly in ebullition here. It is boiling hot in terms of NASH therapeutics. So what we've seen this year is more data about innovative molecules, and some of them, like semaglutide, have been awaited for a long time. That was a long 18-month trial, reported positive results this year. We also have some disappointing results from elafibranor, a PPAR alpha and delta agonist, which reported a phase 3 trial that failed at the interim analysis. But at the same time, we had very promising results of another PPAR agonist, which is a pan-PPAR, lanifibranor, which showed in a much smaller trial of shorter duration, surprisingly good histological results. So a lot of progress along the road with some older molecules that confirmed or not their efficacy, and some newcomers, which are very interesting to look at because they come from classes of drugs that are already well established, and yet for which it is important to understand how much more they bring to the table than the first first-in-class compounds. And for instance, I'm referring to Fxr Agonists, and we had results in regard to that tropifexor, which is one Fxr agonist. We also had very, very promising results with other classes of drugs, such as the FGF21 drugs, and now there are several in development, and some of them look very interesting, and here again, a lot to study, because there are significant differences between these compounds, not only in terms of pharmacological structure, but also in terms of what they have shown to be able to achieve in terms of clinical results. And then close to this, we have aldafermin, which is an FGF19 engineered compound, probably closer to the Fxr agonist than to the FGF21 agonist, but which also continues to provide year after year very interesting phase 2A and phase 2B results from smaller trials, trying to fine-tune the dose, and each time with very, very promising results that are each time confirmed by all these subsequent trials, confirmed biochemical efficacy, and then some additional hints of very strong histological efficacy as well. So a lot of new classes of drugs, different stages in development, very promising results. We now take for granted the fact that these trials can be conducted and completed. But I remember 10 years ago, very few people would think that we could do NASH trials, finding these patients, doing the biopsies, following them long enough, having them to accept an end-of-treatment biopsy in order to measure the histological effect. At that time, very few people thought that that would be possible on a larger scale, more than once for one molecule. Well, it turns out there are a lot of these trials ongoing, and clearly the field has adapted to this medical need and to the specifics of the trials in this population. And we start now, hopefully, to reap the benefit. Now, there have been also some interesting reports in terms of how do we measure success in these trials, regardless of what drug is being used. And this has been published this year, very important papers on the limits of the pathological readings and how we should deal with that, and what are the limitations that we should know about, but also confirmatory data, which is very important, about non-invasive markers and how these could measure therapeutic efficacy. Some of these markers, particularly the ones that are based on imaging, are now becoming mainstream in terms of trial conduct. I'm referring, of course, to all the MRI-based methods to measure liver fat content, or even now slowly starting to emerge the ones measuring fibrosis through elastometry methods. So all this makes for more efficient trials, for endpoints that are better standardized, more quantifiable, and provide early results. All this should help drug development. So I found 2020 particularly rich with this type of data that hopefully will help us move towards the day when we'll have a first drug approved. There were some disappointments. For instance, the fact that the NDA by OCA, the obeticholic acid, was apparently rejected, at least in the first round, by the FDA. That was disappointing because it is unclear exactly why that happened, and in particular, we're all conducting these trials, and we continue doing so with an understanding that if you meet some of these histological endpoints and if you demonstrate the benefit, the way it was framed actually by the agencies themselves, and if you demonstrate that benefit with a sufficient margin of confidence in terms of statistical significance, that should give you the solidity of the data to be used to authorize a drug to be put on the market, or at least on the benefit side, it should give you sufficient data for that. And it turns out the situation is a bit more confusing now, because clearly obeticholic acid had reached the efficacy endpoint with sufficiently large level of evidence, and yet the FDA for the moment rejected that initial NDA. So unclear. We'll need to better understand what it is about, and we'll need to continue working on optimizing the endpoints in the clinical trials so that it satisfies both the regulators, it satisfies the operational requirements of these trials, which can't run for 10 years with 500,000 patients in each arm, and which also satisfies the clinical relevance of what they measure. And at the beginning of the year, we thought all this was sort of agreed upon and already established, and yet we now discover that maybe there's more work to do on that. So I think those are the main things I would take away from 2020, and I really hope that we can make even bigger efforts, progress in 2021, particularly if we can go back to an environment when people can interact more easily, exchange ideas more freely together in physical meetings.
Vlad, thanks for a tremendously rich and broad description that covered a lot of territory and a lot of events, I think, efficiently and very, very quickly.
I would say this is terrific, your summary of 2020. And it reminds me of the parallels with NASH natural history, where we think 2 steps forward and 1 step back. And with drug development, I think we've seen the same thing. We've seen some very positive movement forward, but we've had a couple setbacks along the way. And it just struck me that there was some parallels there. As we look forward to 2021, really the question is, how do you see it playing out? What do you see happening? Assuming COVID is what it is and we get vaccinated, and by the end of the year, maybe the second half of the year, we're meeting in person again, we're back to status quo. How do you see the field moving forward? It doesn't just have to be in drugs, but just, non-invasive testing, regulatory ideas. Where do you think we'll be? AI, artificial intelligence play, just some, putting on your futuristic hat. How do you see 2021 playing out?
Well, I think there will be some work to make truly important advances in biomarkers and someday replacing biopsy for the purpose of both clinical practice and clinical trials. So I see 2021 as a continuation of the previous years with a richer set of data demonstrating how we could use these biomarkers. We understand very well their limitations in the sense that they're being compared to liver biopsy, which itself has limitations. So therefore, you can never reach a perfect value of a biomarker because it's compared to an imperfect standard. That we understand. But there is accumulating evidence in favor still leaping over the biopsy and demonstrating the value of these biomarkers as an indicator of the status of the liver, of the prognosis of the patient. I expect more data in 2021. Particularly important, it will be the demonstration, now that we have these large data sets from phase 3 trials, that patients that are labeled responders based on histology are also the same patients where you can see changes in biomarkers that are meaningful, and therefore by using that, we could define response based on those changes in biomarkers, knowing that they have been sort of correlated with histological changes. So I think that will be an important thing that will happen in 2021 because now we have the data. We have data from OCA, we have Regenerate, we have data also from Resolve It and other trials, semaglutide. So I think 2021 will bring us one step further to using these biomarkers as a parameter that determines response in clinical trials. 2021 will also be a very intense brainstorming year in terms of how we use pathology in clinical trials. So that is traditional pathology and how we optimize this. And all the discussions around this with a number of pathologists, the strategy to read these slides. But also in terms of automated aids to pathology, to traditional optical pathology, and this is where the artificial intelligence type of automated reading will come in. And I'm sure 2021 will bring much more information on that. So a lot of this information comes from people doing specific studies, but most of it actually comes from pharma and sponsors that integrate these methods to their clinical trials. This is what happened with MRI-PDFF, and you see that now it's a well-accepted biomarker. I think it might happen the same with AI, but probably the issue with AI would be to sort of standardize and decide what type of approach you were using. MRI-PDFF is just one unique single tool. AI can take different shapes and forms depending on who is developing it. In 2021, I think that the most urgent things we need to work on, and then, of course, all the studies on natural history, understanding the heterogeneity of the disease, the groups at risk. There's still a lot to do. It's still not entirely clear what we do with people with moderate fibrosis, such as the stage 2, F2 people. Should they be treated? Should they not be treated? Are they truly severe? Which one of those will progress? Those type of studies need to be fine-tuned, refined, and we need to have a much better understanding of this intermediate category of patients. So those will be some of the things, but I certainly cannot think now of all the important things that will occur. And maybe, Steve, you'll make a big discovery in 2021, which everybody will be talking about at the end of the year.
You've mentioned so many things, and as I was sitting here listening to you, I was blown away really by how much work is currently being done in NASH. And it's so much more than just a drug to treat NASH, isn't it? It's identifying the right patients that need to be treated. It's developing the right drug for the right patient at the right time in the spectrum of their liver disease. And it's understanding how we move beyond liver biopsy to something that's simpler and easier to use. It's looking at non-invasive tools to assess efficacy of drugs and even begin to predict long-term outcomes. And so I just want a quick follow-up question. When we talk to Mazen, Mazen Noureddin has done quite a bit of work on MRE, looking at predicting an outcome benefit based on or not an outcome benefit, but based using MRE to predict an outcome, and really following on work done by Alina Allen at Mayo. We had a discussion on his segment where we mentioned that while MRI-PDFF might be used as a marker of therapeutic efficacy in drugs that move liver fat, for instance, some of the FGF21s, resmetirom, thyroid hormone receptor betas, and maybe the FGF19, could MR elastography be used in a similar fashion as MRI-PDFF for people with cirrhosis? In other words, as we study cirrhosis and as we study drugs that impact cirrhosis, could we use MR elastography as a marker of efficacy that could then subsequently be used to predict outcomes?
I think based on the data accumulated so far, it is premature. But of course, all this can change with more studies and more data. The problem we have with MRE is that most longer-term phase 2b and 3 trials, most of them did not use MRE in their study design. So we won't have those data. And unfortunately, those data are very important because those are the ones that would associate MRE with histological changes, improvement or deterioration. So the only studies that have used MRE in trials, are the early phase 2A trials. And those are too short, first of all, to allow a biopsy to be done. And then they're too short to really meaningfully assess fibrosis changes. So I'm afraid that for the moment, we're a little bit blocked in a situation where MRE is not being used and studied at its full potential, at least in clinical trials. And most of these phase 3 trials are already underway. They do not have MRE planned because of the sheer number of patients that are included. It's probably something that's not feasible. So I suspect that it will be a little slower for MRE to reach the same point as MRI-PDFF did. Nonetheless, the idea of having elastometry as a marker, it's happening. It's materializing because you saw the data with FibroScan. It's poor man's elastometry, poor man's MRE, but it still provides data that can somehow be associated with histological changes. So that we have data. For MRE, I might be wrong, but I see this happening a little slower for reasons that I mentioned. And then individual studies done by people like you and me do not have the power and the prospective design to really show that MRE tracks with histological changes. We can have occasional anecdotal small series of patients, but we won't have the power that you have with a well-designed trial where people have this before and after. I don't know. What do you think? Do you think that it's coming of age for use in trials and clinical practice?
Well, I think, as you alluded to, it's a work in evolution, and we need to generate data. And only after we generate data can we analyze the data and really apply where we think it will be best utilized. What struck me from the work that Alina Allen has done is that if you take a cirrhotic, for instance, and you have an MRE of 5 kPa, over 3 years of follow-up, that patient has a 20% risk of decompensating. Alternatively, if the MRE at baseline is 8 kPa, over the same 3-year period, there's a 40% risk of decompensating. So my thought would be, if we set a baseline MRE value of, say, 6, for instance, and we reduce it to 5, did we reduce that rate of theoretical progression of that liver disease? And if we do, we can look, for instance, at the work I presented at AASLD on an interim analysis of the first 123 people participating in the resmetirom trial that were dosed at 100 milligrams. And we saw a 50% reduction in MRI-PDFF in the 16 weeks. And we dropped MR elastography by 0.34, which was highly significant. But I don't know what that number really means. Like, I can look at the PDFF number and I can say reducing by 50%. Theoretically, that has some impact on NASH resolution and maybe even fibrosis improvement. But I don't know what an improvement of 0.34 in MR elastography means. And I think that's where we need to get more granular data in 2021.
I think that, just to say very briefly, I think applying this to cirrhosis would be very interesting. But remember, we have so much data on HVPG and prognosis in cirrhotic patients, and still HVPG did not really make it as an undisputed outcome in cirrhotic trials, despite all the massive data we have. So we need to keep this in mind. But of course, it would be great if MRE would be a good indicator of fibrotic changes and clinical events.
You know, you and I could talk forever about these sorts of things, but maybe we should let Roger and Louise ask a question or chime in as well.
I only actually had one comment because it was a great summary. All my thoughts were, in the trials that were conducted in 2020, all of the participants were already recruited and known for most of those studies. So the ones that have been delayed, the ones that have been postponed, we have waiting lists in liver units and hepatology throughout the UK, and some of them have waiting lists of over 12 months now for patients. So the actual ability to locate patients in 2020 with the length of waiting lists, certainly in the UK and maybe around the world, does that pose a problem to identifying the right patients for the future trials?
How so, Louise? How exactly would that evolve to be a problem?
If you keep to your normal clinic schedules, whereby you're only seeing 30 or 40 patients in a hospital a week through a specialist hepatology department, and that doesn't change, and yet your waiting list is a year long, those 30 or 40 patients coming through may not be of quality for NASH studies. What you want is the pool of a year's worth of patients waiting, screened sufficiently quickly to be able to identify patients to go into the future studies. I don't think the volume of patients is going to come through in the way that it was before, because there was no real waiting list.
But the question is, does this waiting list dilute actually itself in the large pool of patients who are not even recognized and diagnosed? That's a problem. But it is true that COVID impacted heavily in terms of patient management, particularly those for reasons other than COVID. And a lot of people missed their appointments. A lot of people missed the exams that were supposed to detect cancer or monitor for cancer appearance. So it is quite dramatic in terms of negative impact in non-COVID diseases. I think that, at least from my point of view where I practice, I think it's manageable. The biggest problem we face is not so much having too many people waiting, because we adapt ourselves. We work longer, or we have more people that try to help us for that. But the real problem is not enough patients being referred to us, despite them having the risk factors for NASH. So the eternal problem of lack of recognition of the disease. I personally don't see here in France. So there were some delays, but on and off, we've been working like maybe 80 to 90% of our capacity in the past 6 months. So it is a problem, but I think we will be able to manage it. And most of these patients are not like they need to be treated or managed within 1 month, right? You still have enough time to do the work properly and in a good way.
So, Vlad, let me ask a different kind of question. There's a term called a blinding flash of the obvious. What that refers to is something that nobody sees, and then once it becomes clear, you can't believe you didn't always know that. Okay? Can you think of anything in 2020 that would qualify? A moment that when it became clear, it transformed all your thinking both forward and backward? It could be any of the drug trials. I sometimes think about PDFF when I think about this, but where would you go?
If you frame it that way, it's clearly the pathological problem, issue with pathology. With pathologists, with the way you read the slides, with the way you define success in terms of histological efficacy, yeah, clearly. I mean, we kind of didn't want to see the problem, but we could not avoid it once it was out there in our face. And Stephen's work on that and the paper he published together with other people from the Sirius trial were the first ones that clearly show that we have a problem. We know that histology can be variable, but here they've shown that this can have a real impact in the way you define success or failure in a trial. So, and that clearly is not just me, since the FDA now is no longer happy with just the way we did business before, and they're really asking people to think about ways to overcome that. So I would think that this would be the easiest answer to your question, but it's an interesting question. Yeah.
I think back to NASH last January, right? And Stephen presenting the serious results, and then there was a panel with a debate actually with you and Rohit and Stephen. And watching it, and realizing that you all were grappling with this thing that was that important that just hadn't been there before. So that makes a tremendous amount of sense to me. Stephen had to jump off early, and you've been really generous with your time. We're looking forward to having you back with us again in 2021. Thanks for joining today. And Vlad, thanks for doing a tremendous job of summarizing a whole year very cogently and without a lot of time to prep for it.
Thanks for giving us the opportunity also to share with you our point of view and also to provide these podcasts, which are very educational.
Our pleasure. More fun ahead. Or in the words of Sancho Panza, more misadventures.
Merry Christmas and happy vaccine.
Next, we have Mazen Noureddin, who joined us for the AASLD coverage and was one of the key people with Donna Cryer on Global Liver Institute's Beyond the Biopsy initiative in September. Mazen will discuss recent work, much of it his, on the increasing role of non-invasive testing throughout the diagnostic, treatment, and monitoring processes, where we've come, where we might be going. This discussion is with our good friend Mazen Noureddin, who joined us first at AASLD and has worked extensively in a variety of areas and was first mentioned repeatedly on this podcast by Donna Cryer when talking about the Beyond the Biopsy tour that the two of them did together in September. And Stephen, why don't you take maybe a minute and just tee up this conversation for us?
Yeah, absolutely, Roger. Thank you. So amazing. Great to have you on with us today. And thanks for all your seminal work that you've done in the field, particularly in the past year in the era of COVID, where you've come online with a very elegant publication looking at screening of NAFL diabetic patients, and then some of the seminal work you've done looking at non-invasive testing strategies, combinations of both wet and imaging biomarkers. Great to have you on with us today. Love to hear your insights and comments as to where you believe the field has come in 2020, where we're looking forward to in 2021 relative to those particular areas of interest.
Stephen, Roger, and Luis, it's a privilege to be here again today. It's a privilege to get a compliment from you, Stephen. You have led the field nicely and with many therapeutic drugs and other areas in the field. So I guess compared to also when you talked about the FDA endpoints, I want to look or focus on real life reality, what we're going to do in the future for these patients. And many things happen in this 2020, other than the coronavirus, of course. I think we have very good data now giving us some comfort that we don't have to stick our patient right and left, at least for monitoring. So let me break it into 3 pockets, and the screening will be one of these pockets. So pocket number 1 is identifying the patient that are at risk. And since 2015 until now, we have clear data that's showing patients with F2, stage 2 fibrosis and higher, are the highest risk for morbidity and mortality. So in this year, and I'll go to them in a second, a lot of data came out showing confidence that we can tell who's F2 and higher using non-invasive testing. The second interesting pocket nowadays is the concept of NASH in 4 and higher and F2 and higher, which, as you know, this is what we use in clinical trials based on liver biopsy to enter clinical trials. But now we have non-invasive testing that can accurately detect those. We'll go to those in a second. And the last one, which is, I think, very important, the non-invasive testing role in monitoring therapy responsiveness. And this is very promising because, as I said, serial biopsy is not a great option for such a prevalent disease. So let me go back to pocket 1. And I'm glad that a lot of people identify the F2 and higher as a cutoff because it gave us kind of sense of the clinical importance and who should we looking for. So now we have a large pocket of options: transient elastography, MR elastography, serum-based testing such as Fxr, NAFL, fibrosis score, and what we saw in 2020, combining these biomarkers to reach high area under the curve, specificity, sensitivity, and positive predictive value. So as you mentioned earlier, Stephen, we need to screen the high-risk population. And we relied on that work, the F2 data and higher for comorbidity and mortality, and that we have to identify these patients. And then we relied on non-invasive testing to perform a cost-effective analysis. And that cost-effective analysis, what we basically said, compared to the AASLD guidelines in the past that they said there's no cost-effective analysis to show screening is effective, we said, okay, this guidelines relied on a very nice study by Kathleen Corey, but we updated it since then because since her study, the F2 story was not quite well known, and non-invasive testing were not as used. So we said, okay, we'll use the transient elastography to detect F2 patient and higher. And of course, that was cost effective because of the disease prevalence. We looked at the diabetics that they are at highest risk. So they turned to be cost effective. And I hope that will be included in the AASLD guidelines. We also compared to the biopsy in this study, and the biopsy did not come out cost effective as everyone would expect. So it doesn't just have to be transient elastography. It can be any non-invasive biomarker to look for F2 and higher that it's around the same price. So I hope that will be included in the next AASLD guidelines, not as a publication, but rather just a recommendation to help our patients out there. And as you know, a lot of them are hiding in endo clinic and PCP clinics, and they get in trouble without them even knowing at one point. The second pocket, as I mentioned, is also encouraging and very promising is the NASH with NAS4 and higher and F2 and higher. And as you know, FDA requires a biopsy in phase 3 studies to enter these patients' clinical trials. So when they get approved, we want to look for something equivalent non-invasively. And a lot of things have been studied right now. So, Stephen, you have involved in 2 of the major works that have been published between 2019 and 2020, which is the FAST score by the transient elastography machine, which is, as you know, it's a steatosis, fibrosis, and AST, and the NIS4. It's a serum biomarker that includes multiple components that also detect the same thing. We also have done similar work using MRI that you and I are working on, and there are others to come. The last pocket that I want to mention is response to therapy, and I'm not going to take too long, but we have seen a very promising data showing that transient elastography, Fxr, we had ALT before, but now a lot of other biomarkers such as the CT1, CORC3, ELF, many others are going the right direction with the therapy, as well as some data on biopsy, but we need more of that field. And I want to add one last thing, that the prediction to progression to cirrhosis, there were some papers that are evolving. You and I talked about Elena Allen's paper showing that increase on stiffness NMRE correlates with decompensation. She had a very nice longitudinal data for many years. We had a smaller cohort that also showed the same thing, not as detailed as her cohort, but also emphasizing on the same thing, that increased stiffness tells you that this patient probably going to decompensate based on MRE. And that was shown before in FibroScan. And we identified cutoff on MRE, which is 6.48, that correlates with decompensation. I think, to me, this year is a year of hope for patients. I'm sure others will talk about therapies, and there were some promising therapies. So to me, the glass is half full, and with the non-invasive testing and future use of those non-invasive testing, I think we have a good year, not a bad year for NASH compared to the year we had bad overall with the COVID-19. So that's how I see this year, and I'm always optimistic. So I hope that you guys have the same view on the NIT.
Yeah, I get asked all the time, is your glass half full or half empty? My glass is always half full. And your contributions to the field have been very, very important during this year. I just wanted to follow up with 2 comments. Number one, your paper on screening of diabetics for fatty liver, I believe, just a point of clarification, that one of the things you added into your model that Kathleen Corey did not have was liver cancer or liver transplant. I can't remember which one it was. If you could clarify that. The other question or comment more, actually it's a question. I know Naeem Al-Khoury has mentioned this before, and I thought it was really an interesting comment, and I want to get your take on it. And that is, on the context of use of measuring therapeutic efficacy for metabolic drugs, we look at MRI-PDFF, and we see data this past year reflecting that actually the higher you go in relative liver fat content reduction, the better the odds of having NASH resolution and even fibrosis improvement, as outlined in the NGM trial and with resmetirom. Do you see MR elastography potentially filling that role for cirrhotics, using that as a non-invasive tool for a marker of therapeutic efficacy?
Smart and tough question, Stephen. Let me take the first one. It's an easy one because I did the study. So I think the difference between our paper and Dr. Corey's paper, we took advantage of 3 things. Hepatocellular carcinoma, as you mentioned, but also we took advantage of the recent knowledge that she didn't know back then, which is we want to look for F2 and higher patient. In her initial paper, again, knowledge was not known about the F2s. She looked at NASH using biopsy. We said, no, let's look for F2 using non-invasive testing, and we added the hepatocellular carcinoma. A lot of data came from Fxr Agonists on disease transition and prevalence and all this. So I do thank him so much for publishing so much data that we relied on it heavily in that cost effectiveness. For question number 2, let me start with this. I think it's good to say, I don't know. So as you know, that in cirrhotics, you have 2 types of patients and multiple pockets. There are patients still with activity, steatohepatitis. And at certain degree, as you know, steatosis and inflammation decrease. You see that in cirrhotics, but you see it also in the further you go, meaning the worse the disease, the steatohepatitis starts disappearing, which eventually goes to what we call the cryptogenic cirrhosis. And there was a paper by Fxr Agonists in 2019, I think JHAP, came from the simtuzumab data showed that these people that they're quote-unquote cryptogenic, they have a higher collagen content, probably thicker septa and all this, and they decompensate worse. So to answer your question directly, I don't suspect that MRI PDFF will be helpful in this group. Rather, it will be helpful on those that they have steatohepatitis activity, that you can reverse it and you can show that on MRI PDFF or CT1. And I think with time, PDFF will show us more data, and in terms of it's able to predict reversing cirrhosis, I think that will be based on its improvement of steatohepatitis. So that's how I break it. Cirrhotic, cryptogenic, or no activity, MRI PDFF will play lesser role versus those with the steatohepatitis activity, MRI PDFF will play a role.
I don't want to hijack the conversation completely. I know Roger, Louise may have some quick comments or questions. Just to clarify, the work that Alina and you have done on showing that MRE can predict outcomes, I'm just thinking down the road in 2021 and beyond, if we're able to show regression of stiffness on MR elastography in a clinical trial, do you think that tool could be used, MR elastography, to show a reduction in outcomes with a therapeutic modality? And as a result, that would become the endpoint that we could use with the FDA in a cirrhotic trial?
The answer is, this is very thoughtful. I think the answer is yes. We're able to predict this side of the freeway, which is the progression. We need to go the other side of the freeway, which is predicting regression with MRE. And as you know, that this has been slightly difficult with a lot of non-invasive involving trials. And I don't think it's because MRE is not showing that. I think this is an area of research needs to be tapped. And I think there should be some modifications in the way the MRE is read in regression. We can talk about this, and it's a matter of debate, but I think it will eventually, once we find the right way to read MRE regression. We've seen it in transient elastography, more than 25%. So the story there has been nice and needs to be published. But I think MRE has a lot of potential there. We just have to formulate it well.
Thank you.
Thank you. You can tell which one of us lives in Los Angeles by who made the freeway reference as a way of describing where we're going here. I doubt that Louise or I or even Stephen would have gone to a freeway reference, but thank you for that. Louise, you have a question?
Yeah, I have to. It'll be interesting to see. I think everybody knows F2 and above, but Donna was referring in a couple of episodes that the FDA and that have accepted new ICD codes for lower than F2 fibrosis, and whether or not we've got any markers that would be sufficient other than biopsy to pick up the lower grades of NAFLD and NASH fibrosis, whether it's for FibroScan or any of those are sort of sufficient to be able to detect those patients. But the other thing that always strikes me is that we look at cost-effective analysis in screening populations who've already developed disease. And that's a very expensive way to start a cost-effective analysis. And I know there's the Litmus Project, and they're looking through Europe of scanning 30,000 patients throughout Europe with FibroScan in primary care. But does any cost-effective analysis look at the cost of development of the disease before you even start to screen? Because liver disease, we know, and fatty liver disease particularly, has become a colossal global population. We now have 7 out of 10 NCDs as the biggest causes of death worldwide. And it's not a surprise. A lot of them are interlinked with fatty liver disease. So by purely waiting to screen for fatty liver disease, once diabetes, cardiovascular disease, hypertension, high cholesterol is developed, is not ever going to be cost effective. Surely the cost effectivity is screening for the disease to prevent both the fatty liver disease and the associated diseases. Prevention is cost effective. But if we always tail end it, we are never going to produce much evidence. So it was absolutely fantastic and startling that you were able to produce that evidence in those type 2 diabetics over the age of 55, was it?
It was a 55 based on previous models, but we projected it. If we go down to 40, would it cost effective? And it was. So the recommendation was to screen for 40 and higher.
But would it be cost effective to have prevented the diabetes in the first place by screening earlier so that would be my argument. And I come from a liver well-being perspective. If you keep the liver healthy, you develop less of these diseases. And looking earlier changes people's behavior a little bit better than waiting late.
Excellent point. Very well taken. And the question is, why wait until NASH and it's F2 and higher, rather detecting it even with the early stage, even before NASH, or NASH and F0, and try to reverse it? As you know, like a lot of cost-effective analysis data relies on cost of transplant, cost of HCC, which patient population, if you spend that money, it will be, most helpful. So I think that's what we did. I was happy that it's a first step. I do agree with you that looking, for instance, in NASH F0, F1 in a more cost-effective way will be a excellent way to go. So we can start earlier. I can tell you that we considered cardiovascular mortalities, though we need more data there. So we consider both liver-related and cardiovascular. But one way to do it is to go for NASH and F0, F1 as the next step, and through less invasive or less expensive tests and see if they will be cost effective. So I do agree with you. The earlier the start, or you can just argue that obesity, you just start from there. No one should be obese, but that's a different story for a different day. But yeah, I do agree with you, Louise.
Yes, obesity, lean, unless we find the fatty liver throughout the populations, which is difficult to do, as we know, in purely blood tests. I think a lot of the evidence this year about non-invasive tests moving beyond biopsy can provide us with, and particularly people and individuals, the last thing they want to do is undergo a biopsy. It's the biggest ability to make somebody lose weight over 3 months in a hospital I've ever seen. Well, if you don't improve your FibroScan and your CAP score, Dr. X is going to stick a needle through your liver. It changes and it motivates really well. And if you could do that every 3 months, arguably we wouldn't be sitting here with massive pediatric and childhood NAFLD, but and that growing problem. But
I would rather go on a run 3 times a day than doing the liver biopsy every 3 months or 6 months.
For some of them, it's as simple as dropping sugar drinks. It makes such a difference, as you know. But no, it's an entertaining conversation.
So I've got one question, and it goes back to the issue of elastography and PDFF. That when you do, I'm assuming when you do the cost effectiveness of screening, that part of the equation is benefit, part of the equation is cost, and that the cost of elastography is so much lower than the cost of PDFF if you have to use an MRI to do it, that becomes a factor in estimating the value of the two different results. Am I guessing right, or am I guessing wrong on that?
I don't remember the exact price, but this is a point that I dealt with it earlier in my career. 2013 paper, I was a fellow with Rohit Lomba. It was a junior faculty, I was a fellow, and we wrote this hepatology paper on using PDFF as the primary outcome, as one of the outcomes in the NASH clinical trials. And as you know, Rohit did a million papers in this field. But one of the earliest pushbacks and encouragement we had is the price of MRI. Institutions push for the highest price. But if you really look at Medicare price, both of them, now MRE has a new CPT code. I don't remember the exact coding, but both of them are 3 and 500, especially if you get limited MRI abdomen. So they are close on price, but they are being sold as expensive. Now, would they be effective for screening with the 3 and 500? I don't think so, because the disease is so prevalent. But would they be cost saving in monitoring the disease compared to biopsy or even diagnosis? We're not talking of screening now. I think so.
So the follow-up thought, right, is that in the next year, 3 years, 5 years, certainly, we will start to see the advent of small technology that is capable of measuring both at reasonably strong, robust levels of reliability. And I'm thinking that that flips the whole equation of when do you screen and what do you screen for, and how robustly do we start to collect PDFF data as a pivotal endpoint, because all of a sudden the economics and the convenience of doing it change dramatically. That's just my hunch. But does that make sense?
I agree. And you can screen with something, let's say Fxr, I'm just calling it ultrasound elastography as we publish. And then once they get to the hepatologist office, they can start with PDFF, hopefully with a reasonable price, if, we all push for it, and follow up with it on a long term. Now we can monitor treatment response with it. Can you do that with MRE? And more data will come. So that's how I see it.
Can I just ask a question on that? With COVID having delayed the assessment of all patients coming through to specialists, is this now an opportunity to, in the real world, move to more non-invasive screening to get those lists down in proper environments, and then, as you were suggesting, use MRE and PDFF fraction and biopsy only when needed? Because we have waiting lists for FibroScan in trusts and hospitals here that are now in excess of 12 months. So during those lists, there are people with undiagnosed cancers. There are obviously people with pre-cirrhotic who will develop cirrhosis, people with cirrhosis who will decompensate, who may not get assessed. So is COVID an opportunity for healthcare now to start looking at innovative technologies to start and using Fxr a bit more, and the serial tests that came out so well at Digital Arnold?
Louise, while you were talking, that you're proposing these great thoughts, I was thinking about 2030 and how the world looked different from NASH standpoint. We were forced to change things. I'm on video all day talking to patients. We're sending phlebotomy to their houses, either biopsy or imaging tests such as MRE. You still have to come to facility. The reason why I'm saying is 2030 is like, I wonder if it's going to be all video. I still miss the handshake with the patient, still prefer it, but video, blood tests at home, and a machine that will go to their home and test them. Or we can be prepared for that during hard times. Hopefully we're not going to have that again, but great thoughts. Yeah, I think we have been very innovative during this time, and your thought is provocative, and I think having home testing with imaging and blood tests can be part of the future.
This has been a fantastic half hour. Thank you so much for joining us, Louise, Stephen, and we'll be spending more time with you, I hope, in 2021 as you come on and we talk about more provocative thoughts and ideas.
I know, and I'm getting my vaccine this month.
Good for you. Good for you. Some of us are jealous. Thank you so much. Talk to you soon. Our third discussion this week is with Stephen Harrison, who certainly needs no introduction on this podcast or anywhere else. Stephen made two contributions. First, he chose to insert questions where appropriate, extending beyond simply thinking about NAFLD and NASH, and to ask questions about cirrhosis and what some of the things we talk about might imply for ultimately successful treatment of cirrhotic patients. For his conversation, he went back to the episode we ran after the Intercept Complete Response letter to provide an update on the role of clinical endpoints in the approval process. Along the way, he'll discuss the evolving use of artificial intelligence in research and new insights on how to get better biopsy scoring using human pathologists, all of whom are expert. So this portion of our year-end wrap-up is with Stephen Harrison, who's been with us, as you know, 40 of these 42 episodes as a co-host and exceptional contributor. The topic we're going to talk about in the next segment is clinical trial endpoints, something we discussed earlier in the year, and Stephen wanted to bring us up to date on what we've learned since June when we discussed it. Stephen, why don't you take 5-6 minutes and lay out the issue for us? Louise and I will have a couple of questions, and then we'll wrap up this segment.
Thanks, Roger. It's been a great pleasure to be a part of this podcast in 2020, and one of the bright spots in an otherwise dreary 2020 with COVID. So one of our previous episodes, we had a discussion about clinical trial endpoints. Brought up that maybe clinical trial endpoints were changed in the sense that the goalposts potentially were moved, and that surrogate approval is likely on the chopping block relative to the CRL that was sent to Intercept. So I'd like to take a few minutes and provide an end of the year 2020 update on where I believe things currently stand. Based on my ongoing conversations with the regulatory authorities, as well as with many pharmaceutical companies in drug development for NASH, I do not believe that the goalposts have changed. Clinical trial endpoints for surrogate approval still revolve around the two histopathologic endpoints. One, NASH resolution without worsening of fibrosis, or two, fibrosis improvement without worsening of NASH. In addition, surrogate conditional approval is still on the table and has not been taken off. So I think that point needs to be made clear. Having said that, where I think we move to in 2021 is an aggressive, conscientious effort to begin to improve on the current endpoints because of all the noise that's currently surrounding them. And potentially, this would involve a couple of different techniques. Number one, given the way we currently evaluate histopathology, it would be nice if we could have a uniform way in which we collect the liver biopsy sample, process it, and analyze it. Number two, I think we've learned a lot about the complexities of statistical analyses relative to inter and intra-observer variability. As a result of this, I think we begin to move towards a better way to analyze the output provided by the histopathology. One way that I think that can happen is by ensuring that we have two pathologists independently read the paired liver biopsy samples in a blinded format, then have them report their data, and where they differ, they come together, look at the slide, adjudicate their differences, and develop a consensus opinion. One thing that I think would also be helpful is prior to ever looking at the baseline data, the two pathologists could come together and harmonize their thought by reviewing multiple cases, particularly the borderline cases, to gain agreement on what constitutes NASH and fibrosis would be incredibly helpful. Subsequently, I think additional benefits would begin to utilize artificial intelligence to overlay with what the hepatopathologist is calling, so that we can begin to provide more fully quantitative evaluation of changes in histopathology and provide more clarity around how drugs are working relative to liver biopsies. Ultimately, I think we use artificial intelligence as a bridge to non-invasive testing platforms. And I think through collaborative efforts such as Litmus, Nimble, the Liver Forum, key opinion leaders, patient advocacy-based groups, and pharmaceutical companies, we will be able to achieve these goals. So the future looks bright. We've had a great year of readouts in the NASH field. Many presentations were provided at the Digital ILC and Digital AASLD, giving us great promise for drug development in this field. As we move towards better endpoint analyses, we will gain more clarity on how effective these drugs can be, and ultimately their ability to predict long-term patient outcome with surrogate endpoints. So I'll stop there and see if you have any questions about my thoughts.
I thought they were excellent points, and I think obviously there has been a lot of questioning for sort of surrogate endpoints during this year, but it sounds as if you're sort of calling for a very central set of histopathologists to review all NAFLD and NASH studies. Would that be right? So that it was consistent in each region of the world, for example, so that every study used the same histopathologist. That's about the only way that you could level that playing field? Is that what I was hearing?
Well, maybe let me clarify one or 2 points. If you look at clinicaltrials.gov and you look under the heading of NASH, there are currently 34 trials in clinicaltrials.gov, and only more will be added to that number. Collectively, we're talking around 11,000 F2 and F3 NASH patients. That's a lot of work for just a handful of pathologists to review. I do think we are limited by the number of experienced expert hepatopathologists that can render an opinion of this magnitude for NASH clinical trial development. And I do think we need to grow the bench in bringing on newer pathologists. However, if we look at this very pragmatically. There are about 6 to 7 pathologists around the country and around the world that predominantly read the clinical trials that are currently in development. In fact, maybe even 3 or 4 of them have predominantly read the majority of them. So for the foreseeable near future, I think we need to begin to just do some very pragmatic things. Number one, we just need to all do it the same way every time. And I think ultimately where we have 2 pathologists that can come together, harmonize their thoughts, review the data independently and adjudicate their differences, that gives us the best shot on goal in the short term. In the long term, we need to begin to transition away to non-invasive testing, given the limitations of histopathology, with the, all the limitations that we understand relative to that. And maybe one that we don't always talk about, and that is the limitation of expert pathologists to read the data. That's another key point that you highlight, Louise, that I think is worth noting, because we always talk about the noise of the biopsy, the heterogeneity of the biopsy, the sampling variability, the discomfort associated with it. But we don't always talk about the lack of expert pathologists in reading those slides. And I think that is another point to consider when we begin to talk about this transition in endpoints away from a semi-quantitative analysis to a fully quantitative assessment to a non-invasive assessment.
Stephen, does that mean that a move to using AI as the predominant mode for reading would enable us to process trials quicker? If you've got 6 or 7 people in 11,000 cases, that feels like a bottleneck.
Well, it is a bottleneck, Roger. And I think, the agency is taking very deliberate steps, very carefully considering how we can begin to overlay AI onto semi-quantitative assessment of collagen and the other aspects of the NAFLD activity score. From my understanding, that kind of begins with an artificial intelligence readout of the liver slide, and then a pathologist looking at that with the corresponding H&E and trichrome and saying yes or no, the computer got it right, the computer didn't get it right. And I think as we show, hopefully, that the computer is getting it right 98, 99% of the time, that eventually we can transition away from the need for that overlay read by the pathologist, and we can have simply the AI read. Once we get to that point, we no longer need to put so much emphasis on just a handful of pathopathologists reading the data. So maybe that's how we get there.
I have 3 questions left if we have time to do all of them. First one is, how long do you expect that transition will take? How long to get to the point where we have AI and a pathologist, and then beyond that to AI doing the read by itself?
If you look at the way the agency is working with the groups within NASH to get to that point, you have to have a drug development tool such as AI. You have to present that data to the agency in the form of an LOI. It gets approved. Then you put together a formal development plan to the agency. You have to generate the data from that plan, analyze the plan, analyze the results from that data, and share it with the agency. And all that takes time. I would say in the most expeditious timeline, we're probably a year and a half out from having that initial overlay of AI on top of the pathologist reading. And beyond that, we have to get comfortable with the AI output, and that's probably another year. So we're probably 2.5 years out at the earliest before AI, if at all, would replace a semi-quantitative assessment.
So all this came up in the context of skepticism around OCA and whether OCA was going to deliver clinical outcomes. What leads you to believe that clinical outcomes might not be necessary?
Ultimately, clinical outcomes are necessary, but in the vein of conditional approval or surrogate approval, there is a lot of data being generated that's incredibly positive that these tests are predictive. And again, I mentioned this on a separate podcast, but I would invite everybody to review the paper written by Alina Allen on MR elastography published in Clinical Gastroenterology and Hepatology, where she showed in the setting of cirrhosis that if you have an MRE of 5 kPa, that over a 3-year time period, it predicted decompensation to the tune of about 20% of the patients. Alternatively, if your MRE was 8 kPa, over that same 3-year period, 40% of the patients decompensated. As more and more of that data comes out, it will inform clinical trials. It'll inform regulatory authorities, and potentially it'll help us pivot away from our current semi-quantitative or fully quantitative liver biopsy analysis to what people affectionately call a surrogate of a surrogate. I'm very confident that we can get there.
Louise, closing question. Stephen, closing thought.
My only question was, how do you see 2020 going down in the sort of natural history of development of NAFLD and NASH treatments and outcomes?
Well, I think people will look back on 2020 with a lot of different thoughts, and NASH drug development probably won't be at the top of their list of thoughts relative to what happened in 2020. However, for those of us that live in this space, it was a critical juncture, a very important year where we had hidden in all of the pandemic discussion incredibly good results come out of early phase trials in NASH. I think it highlights the fact that the more we learn about NASH, the better our therapeutic modalities become, and we're beginning to see fruits of that labor. Now, the focus has also been on this whole endpoint discussion because it became clear that the current endpoints, as they are defined, are quite noisy. And drugs from the first generation of pipeline that went through really hit that noisy endpoint hard, and it was hard for them to overcome the endpoint. I think with the second and third generation drugs pushing through to paired liver biopsy endpoints, we're going to begin to see them rise above that noise. But where we're able to shrink the noise, we'll be able to further accentuate the potential benefit of the drug, and I think give us more clarity on exactly what mechanisms are best. And as we begin to look at combination therapy, which ones we should begin to combine to augment and accentuate both of them working together.
I think that sounds like a great note to wrap up on, Stephen. Thanks for your time, and we'll see you on the podcast. The final piece of this episode is from me. I discuss how a disease class whose stakeholders were devastated by bad news in the middle of the year comes to the end of the year so much more confident and robust, and how the complete response letter for obeticholic acid may have spurred activities leading to the optimism so many feel today. I respect Intercept's role of leadership in the class. I mean, this is no comment on that company. They've been great. But the year evolved in the way it did differently in the wake of the CRL. And that might not be all bad. With that, let's shift to the second half of this, which is mine. As you all know, well, you both know, and people who listen know, I'm a big fan of dynamic systems theory. And I think 2020 in fatty liver disease has been a rather interesting example of several things that lie around all these ideas of dynamic systems and complexity and chaos. Well, let me start at NASH-TAG in 2019, when I first became really acquainted with the fatty liver community. And basically, people were trying to figure out how to get better biopsy scores for drugs. And then I'll go forward a year, a little more on the disease, but most compellingly, a paper that Stephen presented on the inter and intra-reader reliability issues surrounding biopsy. And coupled with the idea that we were starting to see more papers on MRI-PDFF and that we had some non-invasive tests coming to market, you could see the beginnings of a momentum, even before you get to omics, to think differently about how you test and evaluate. One of the comments I made in 2019 was that it seemed to me that NASH was a horribly imprecise definition. I mean, I remember 50 years ago, Nixon announced we were going to end the war on cancer, and that started with the presumption that there was something called cancer and there was some way to deal with it. And then it turned out that different cancers were different. And there was something called breast cancer, for example. But then it turned out there were 6 things called breast cancer. And over time, the complexity of the human body overwhelmed the simple definitions that people started with. Here, all you had to do was take a look at the idea that you could get lean NASH, overweight NASH, and obese NASH, and they didn't seem to be the same thing. I was saying this to Stephen when we first met, that immediately struck me as the idea there was no such thing as NASH, or no one thing called NASH, or MASH. I don't care what you call it, there wasn't one of them. What's interesting is that healthcare markets invest in pursuit of drugs, because drugs are what drive changes in markets. And there was a generalized belief that 2020 would be the year that drugs came to market, at least one drug. People were pretty confident about OCA. People were shaky about elafibranor, but pretty confident about OCA. And a lot of investment horizon money, not only in terms of more drugs, but in terms of diagnostics to be there when there was a drug on the market, and education, all that money got spent in the belief that this market was going to be ready in 2020. And when OCA got its CRL, when Intercept received the CRL, there was a rather immediate panic that all the investment money would dry up and that would slow the path of knowledge. But what I believe we've observed is that there was so much momentum around the idea that there was going to be a drug that the spending done around that and the conclusions and the lessons that it gave us have continued. We know a lot more now, and conversations resulting from a variety of things, including Stephen's paper, have led the agencies, and specifically FDA, to go back to the question of is biopsy the right way to proceed. At the same time that that's going on, armed with the knowledge that you need non-invasive testing if this was ever going to go into the population, people who are doing drug development started looking at more things beyond testing, and the people who are doing diagnostic development upped their investments. So we get this positive momentum of knowledge, and we get people investing in omics and things that are downstream, based to some degree, I think, on a belief as to when the market was going to have drugs in it, and as a result, that explosion was going to take off. And what we wind up having this year is this real explosion of knowledge, very, very exciting stuff, and no drug yet, which isn't usually how it works. But it might be better, because the other thing that drugs do is the first drug that launches spends the money educating physicians on the disease that they're able to treat, which may not be the best definition of the disease. And I think there's an argument that goes that would have been true for OCA. The more knowledge we develop before we have to lock in on what the drugs are supposed to do, I think the better shape the market takes. So one way to look at the last 18 months is a lot of twists and turns, but in the end, a broader, deeper, more objective knowledge base about what the disease is that collectively constitute NASH, MASH, or whatever you want to call it, are. And as long as funds continue to be available and good drugs are still being developed, and that's the way it feels from here, that's probably a pretty good outcome for everybody except, and Louise, I can't ignore this, and Donna would jump on me if she were here, patients who are looking for hope and who are looking for therapies they needed. So knowledge does better. Maybe the patients don't do as well. But I don't think it's the dynamic anybody would have predicted at the beginning of the year. And I just find it fascinating and, in fact, optimistic and encouraging in many ways, with the caveat about the patients.
Yeah, I think that's fantastic. That's a very thoughtful summary of where we were and have come in 2020. And as we begin to turn the page to 2021, how interesting the parallels between what we just talked about with the confusion around nomenclature and the parallel confusion around drug development. And I think the same rules apply, right? What we've seen is a very careful, analytical evaluation by the FDA, and the FDA said it's not enough. The placebo-adjusted response rate of 11% with the risk profile that the drug presents is, as it currently stands, not enough to get approval. They didn't say won't get approval. They just said, let's put some more thought into this. And that's what's happened across the board. And as we realize that drugs that target specific inflammatory processes while working in an animal model appear to be redundant in humans and don't work as well as a result of that. And maybe the same thing with fibrosis. What we've learned is to be smarter about the pathways that we target. And I think that's what we see with the second wave of drugs. And I use second in a very broad term, because these newer generation FXRs potentially, while still having some of the positive effects, are able to mitigate some of the negative effects. There was a press release by Medicrin that just came out looking at their second generation FXR that I think is informative. The FGF21s are not all created equal. We know that if you have balanced potency between FGF1, 2, and 3C, that you're likely to have a differential impact on underlying liver disease. We're seeing work now where companies are taking the different enantiomers of a compound and isolating the enantiomer that is actually having the positive effect and minimizing the enantiomer that is associated with adverse events. We've seen that with Poxel. We saw Sirius try to do that with their MSDC0602K, where they're trying to get away from direct activation of PPAR gamma like pioglitazone by modulating the mitochondrial pyruvate carrier complex. And in so doing that, can we accentuate the positive impact of activation of the nuclear hormone receptor PPAR, but not carry the AE profile? So all this is slowly marching forward. And interestingly, to your comment, Roger, it will benefit the patient so much more in the long run, rather than just rushing a drug to the front lines. That's the same concept that's been raised with the vaccinations for COVID-19. Let's don't short circuit the process. Let's don't take shortcuts. Let's efficiently and effectively, using good science and all the rules and boundaries that exist within development of a vaccine, let's just do it, but do it with the collaboration of all all so that we can cut the red tape, we can limit bureaucracy, and we can achieve success in as little as 10 months, which is what we see happening with the Pfizer and the Moderna vaccination. And I think that same thought process is beginning to be applied to fatty liver disease, where we're not just rushing into this thing, but we're trying to deliberately look at all of the blind spots and develop drugs that are not only going to function on improving liver disease, but have pleiotropic effects on the whole metabolic milieu of dysfunction that exists in these people, so that we impact not only liver health, but cardiovascular health and malignancy prevention and all the things that kill these people. We've made huge advances in 2020, despite all the pandemic issues that we've been faced with. Has it been easy? No, it hasn't. It's been a great challenge, but as a community, we've risen to that challenge, and I'm excited to see where we go in 2021 as a result of that.
Yeah, I agree with all of that. You know, the funny thing is, a lot of the processes that you were describing usually happen in a drug market after agents come. So people start looking for enantiomers, for example, after the first molecule in a class gets into the market and it's sloppy and it's messy, but it gets approved. Then people start figuring out how to, what do I do to this to make it more positive, less negative? What's interesting here is all that's going on based on failures, not successes. And I think that is interesting. I think to some degree it has to do with the idea, that, as I say, that people thought that OCA would get approved and that would jump the market, and that would mean that there was more money coming faster. I do agree with you. It's probably better for the patient in the long run, as long as the patient can manage her or his liver long enough to get there. But this becomes a different dynamic, exciting dynamic for that reason. And you're right. What's happened in the last year has just been rather remarkable.
I think as a sense of following the OCA sort of setback, I think patient groups and patients are speaking up. They're trying to get involved a little bit more. Nothing without us, nothing about us without us. But I also think the new waves of drugs that are coming that target, as Stephen and everybody allude to, is the cardiovascular side, the diabetes, and the other metabolic components. A lot of these patients and people, not only do their physicians not look for the liver component in that patient, even when they've had their first MI or their stroke or they've developed type 2 diabetes. But if we can get a drug that targets those diseases and targets the liver disease underneath, whether it's noted or not, there has to be a benefit. And with non-communicable diseases now making 7 out of 10 of the top biggest killers in the world, if you have a multi-effect medication that affects certainly the biggest 3 or 4 of those killers outside smoking, then that itself is a potential massive win, not only for patients, but for low and middle income countries who are the biggest adversely affected populations and indigenous populations in these diseases. So the potential long-term win for multimorbidity management, I suppose, and conditions may well have started to really be visualized, I suppose, this year. And that's very exciting.
I agree. I also wonder if it isn't a little complicated. I think about semaglutide showing significant impact on steatosis and nothing on fibrosis. And you would think that the GLP-1s, and particularly semaglutide, would be exactly the kind of agent you just described. But, and Stephen, I'll ask you this question. If it doesn't affect fibrosis, what does that tell us about its role in an integrated multi-metabolic syndrome process?
If it does nothing on fibrosis, I think it makes me rethink the whole paradigm of improving fat in the liver and the liver then healing itself. And I want to be very careful that we don't make that leap just yet. If we go back and look at the data, there was only mid-50s patients in each study group. 3 or 4 patients in each group would swing that to a positive impact on fibrosis. The other thing to consider is artificial intelligence. Fully quantitative assessment of collagen has not been applied to that data set yet. And we've seen every case where AI was applied and fully quantitative assessment of collagen was done, that the actual impact of drug was accentuated in every case. So I'm unwilling at this point to change my paradigm of thinking, which is if I take away the insult to the liver that's causing all of the inflammatory cascade and activation of stellate cells, that the liver will get better. I'm not willing to shift off of that yet, because in Hep C, if I cure it, if I suppress B, if I quit drinking alcohol, if I treat the autoimmune, every single time the liver gets better. So either fat's not the issue, or we haven't evaluated this study set well enough to really know what's happening to fibrosis. And I think it's the latter.
So I'm hoping it's the latter, because if it's the former, then all this gets a lot more complicated again, right? I think we'd all hope for the latter to be true, that better analysis would demonstrate that the paradigm holds.
Yeah, absolutely. We just need to, I suppose, get more people cured or prevented. You know, I'm more about liver health now, in the context that if we teach them to look after their liver or even know where it is, then we can move forward. And we need both. We need the drugs and the will to be able to cure and locate, and we need people to actually not get themselves there either deliberately or by accident, I suppose. If this is not a tsunami, it's bigger than a tsunami with the figures that are forecast over the next few years, and particularly in children and adolescents. It's something we have to do something about. For me, we're not going to address non-communicable diseases if we ignore the liver. It's a vital component of that. And any of these medications, even if you were to target cardiovascular disease, and I agree with Stephen, I think if you remove liver fat, we will see the liver eventually start to heal itself if we don't put another insult on it. So it's an amazing organ, that's all I can say.
Yeah, it is. I think we agree. So, Stephen, you have anything you want to add, or I'll just put a bow on this and then we can go on?
I think less is more. So I'm going to let you wrap it up.
Can I make one more comment that you may or may not choose to use? But actually, 2020 is the International Year of the Nurse. And obviously it was due to the birth of Florence Nightingale, who set up modern nursing, and she was a statistician, as we know. And in fact, she was the first woman accepted into the Royal College of Statisticians. I'd like to give a great shout out to all of the liver nurses and physicians who've been moved out of hepatology to support the COVID outbreak and really step right up and be counted in the International Year of the Nurse. The nurses and doctors have been absolutely key throughout the world. So big thank you to everybody.
I couldn't agree more.
Well stated.
And I think we're going to let that be the wrap-up note on this conversation, even though it isn't what the conversation was about. It's been a remarkable year. I think Stephen did make the point, which I agree with, that if you knew at the beginning of the year that you were going to have COVID and you were going to have OCA not get approved and a couple of other things, you would not have expected this much optimism around this disease at the end of the year. But I think, Louise, as I guess Mazen did in his conversation also, we need to put all that optimism in the context of everything else this year has been. And yes, nurses and doctors have been fantastic. They've been heroes. I was saying, I think, Stephen, on one of the conversations you weren't on yesterday, I was reading that there's a tremendous uptick in medical school enrollment, because just like after 9/11, more people wanted to join the military because they knew what they wanted to fight. More students now want to go to med school because they want to be on the front lines for the next pandemic, and nursing school as well. And I think that speaks to how inspiring the doctors and nurses around the world have been in the face of all this horror.
Yeah, I agree. It's amazing.
All right. Let me go on. I'm going to start putting fronts and the ends on all of this. So that wraps up our Drugs and Diagnostics episode as part of our year-end review. I hope you found the interviews worthwhile. If there's a particular interview you want to rehear or share with friends, we will be posting each of these individually starting over the weekend, so keep checking for the one you want. We will be back next week with episode 44, Patient Treatments and Perspectives. Until then, stay safe and surf on. Wherever you are, whatever holiday you celebrate, and whatever pandemic constraints you're operating under, enjoy yourself and stay safe. See you next week on the podcast. Bye-bye now.
Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, patient advocate Donna Cryer, liver wellness advocate Louise Campbell, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on the Surfing the NASH Tsunami podcast.